Hi, everybody. Welcome to our webinar on the graph genomes and their application to genomic analysis. I'm joined by my colleague, Seherat Stetikol, who leads the product development for graph genomes at Seven Bridges. And I, Amit Jain, lead the technology development for the same. And together we will guide you through the various results and details of our genome graph implementations, and also give you a live demo. I think before we start, we should maybe give 1 minute for everybody to join. And after that, Sarat will start with an introduction to the graph genomes, and then I will present some in-depth analysis and results, and then we will wrap up with the demo. Yeah, let's hang tight for about a minute, and then we'll kick off. Thank you. Okay, I think we can start now, Sarat. Hi, Sarat. Maybe you're muted. Sorry, guys. I need to find what happened to Sarat. Hopefully, he's just muted and doesn't realize it. Let me give him a shout. Okay, if he doesn't get back, join us in 1 minute. I will carry on. Okay, so I guess it falls upon me to do the introduction. So yeah, let's start the webinar. I will share my screen. Okay, so let's start with a simple introduction of what are graph genomes and how we have implemented them at Seven Bridges. The basic idea of graph genome is that the standard reference haplotype is augmented with genomic variation so that it provides a richer scaffold for mapping reads. And you see the schematic on the right, which is showing this in action. And and when we say that genomic variation of interest, the variation could be a deletion, wherein certain bases in the reference are missing from the sample genome. It could be an insertion where the extra bases are present, which are not present in the reference, or it could be substitutions. And combining all of these and going into the size of these variations where they start affecting the structure of the genome, you have structural variations. So this is how a graph genome is organized. And in addition, we have our implementation of the graph genomes is not limited to only keeping the sequence information, but we can also store relevant information, for example, the frequency with which these variations occur in the population, linkage, and so on. So in order to inform the subsequent analysis and provide better prior information. So what are the advantages that are available to us using this structure versus the standard linear reference? So first of all, there is the issue of when you have a single haplotype reference assembly, there would be some genomes which are proximate to that assembly, maybe because the population from which the assembly was constructed was the population from which they are drawn, and that implies that the people from other populations would not have their genomes as well represented, and they would give rise to more variation. So that introduces a bias. It's a well-known term, reference bias. Secondly, when we have the graph genomes, we can always augment them as a newer and richer information becomes available, as is happening now with the long-read technology, giving us a lot more reliable structural variation. Then third point, and I think this is probably quite important, is that there doesn't need to be only one graph reference. We can tailor them to the intended use case. So for example, you could have a separate graph reference for analyzing individuals drawn from a specific population, or you could have a graph reference for a family to help with rare variant detection, and so on. And we'll see examples of all of these later on in this webinar. Also, graph references are efficient and robust. And here I'm speaking of our implementation, and we have shown that that this extra information in the reference can be handled in a cost-effective and robust way. And again, we'll cover this further down. So here is an example of how graph genomes alleviate reference bias. And this is a schematic, but you are seeing that there are some sequencing reads here, which presumably come from an individual whose genome has extra bases inserted. So you see the gap in the linear reference below, in the linear sequence below the reference, and this is how you expect these reads to look like. And on the right panel, you see what happens when we provide the extra, the knowledge of insertion to the software that is mapping the reads. So and and uh, and it's clear that if you get the configuration towards the right, it is a lot more convenient for calling variants and so on. So following this idea, we developed an implementation of the graph references, and we have constantly been in the forefront of organizations who are developing and popularizing graph references. And we have, for the first time, we made our workflow available to all users on our platforms for free, without paying anything additional. And we have several platforms. Some are academic platforms, for example, CGC, Kavatica, NHLBI, and we also have a commercial platform. So if you are signed up on any of those platforms, you can access our graph tools without having to pay anything extra or to any separate registration or anything like that. So before I dive into the nitty-gritty of what the graphs are and what the numbers look like when we use them and so on, it's worth mentioning that in the recently concluded Precision FTA Truth Challenge, our results from our graph pipeline were judged to be the most accurate in the MHC region when only the short reads are available. This is, we think this is quite significant, and especially given the importance of this region for various applications. And since these are highly polymorphic and difficult to map regions, it's clear that the graphs bring superior, graphs improve the ability to accurately characterize the variants in this region, and this has a lot of applications, including for organ transplant and so on. So now this is my bit, so I should be more confident here. So it's a case study. I will start with showing you some real examples of how graphs are constructed and how and the effects they have. So to start with, we know that the newest version of standard reference, GRCH 38, it includes several alternate contigs. And these alternate contigs are an acknowledgement that single haplotype references are insufficient. So in several regions, instead of having one sequence, you have multiple sequences, which are all which all occur with high frequency in human population. So here we are focusing on one such alternate sequence. The name is chromosome 19, GL38, etc. And this is supposed to be an alternate for the region given here in chromosome 19, 34 million base pairs, etc. So when we look at this alternate sequence, we use it to start the process of constructing a graph reference. And the way we do it is we just map it using the mapping information that GRCH also provides. And we get something like this. We see that this sequence is homologous to the reference for the most part, except for some small sections, all of which become edges in our graph. And for comparison, the bottom figure shows you the population reference, the population graph, just to show the information contained in our entire graph and how this is but a small part of the total information. And the highlighted green sequence here, that is actually an insertion with respect to the chromosome 19 sequence, because it occurs only in the alternate contig. And on the next slide, we will see a sample, which is our standard benchmark sample at G001, mapped against only that green section using the graph, which is the figure on the top. And you can immediately see that there is evidence for this alternate section being present in the sample. It is present as like a homozygous alternate. And when we see the mappings from BWA, then it is a little more complicated, because this part of the genome is actually present in the alt contig, and we see the mappings in the alt, the bottom of the panel, and we see the corresponding section in the reference in the chromosome 19. And so several things are presented here. One is that the chromosome 19, even though this is a homozygous insertion, we still get about 25 reads which span this location, which is not correct, and it might confuse the variant caller. The second is by default, most variant callers will not call anything here. You need to do some post-processing, and even after that post-processing, combining the signal from two places leads to inexact variant calls. So, but with the graph approach, we added that sequence as edges to the graph, and we don't even have to think about it. No post-processing, nothing. You analyze the sample and you get an insertion called there corresponding to that alternate. So we think that's a big advantage. And this was the starting point. So, and as you saw, we also add other variations drawn from the population to this graph. And we have published a population graph which is constructed following the scheme that I just showed you. And this slide just covers the various data sources that went into constructing that graph. And the data sources that we have are the 1000 Genomes project, Simon's diversity, HapMap, etc., for population variants drawn from pretty much all around the globe. That is what the panels on the right show. And we also have some structural indels. They come from Mills indels and longer variants from the alternate contigs. And we further extended this. So let's first look at the results from this graph. And we see that the figures here are comparing the F1 scores. F1 scores measure accuracy. They penalize missed variants and false variants equally. So they are a pretty good and pretty standard measure of measuring pipeline accuracy. And when we look at the figures here, the blue bars are for the graph genome workflow. The red is PWA, and again, other workflows that work off PWA mappings. And we see that first thing to note is there are two samples here. One is a standard benchmark sample, HG002, and we see that all pipelines are pretty good at calling both SNPs and indels in that standard benchmark sample. So, and to the extent that, at least for SNPs, there's pretty hard to distinguish. We are really yak shaving here. So, sorry. So we also, for workflow development, we also like to use another sample, which is the SynDip mixture published by Heng Li. It is constructed from, it's a synthetic benchmark constructed by mixing reads from two homozygous hereditary form mole samples. And it has a much longer range of variations and also longer high confidence regions. And it lets you meaningfully differentiate between the various pipelines, variant calling accuracy. So if you look at the numbers, for SNPs, most pipelines are pretty much of the same order on the standard benchmark. For indels, they are a bit more differentiated, and this distinction is more noticeable on the synthetic benchmark. And in both these benchmarks, we see that graph pipeline is near the top, and it is noticeably more accurate in calling indels. And this is the effect of having the prior information about indels included in your reference. So the reads map better, as we saw in examples earlier. And we extended this, the graph that I was showing you earlier, to also include structural variants. And here we see a measurement of structural variant calling accuracy. Recently, Genome in a Bottle Consortium published SVs for the same HG002 sample. And they are plotted here as the black lines following the number of variants for each variant size. And we compare them by seeing the calls made by the graph genome, which is the green line, and the standard structural variant caller, which is MANTA, and that's the orange line. So if we look at deletions, which are towards the left, you see that MANTA calls more deletions than the graph, the green line, but graph calls more insertions than MANTA. But we did another experiment when, after adding these SPs to the graph, we see that its recall of these SPs improves dramatically for both insertions and deletions. And further, when we run MANTA on the graph-generated BAM files, then we see that MANTA's recall also improves. And that could only be because the read mappings are better, right? Because MANTA doesn't know about extra information, graph-related information that we keep in the BAM file. So, yeah, and we hope that this we will be able to position the graph pipeline as the first unified pipeline capable of calling short variants as well as SVs. And finally, the performance implications of these extra capabilities. So Graph does a lot more work. It maps reads against a much richer scaffold, so it has to do more comparisons. So you would expect that this would be reflected in the performance and the runtime. But our implementation has, from the very beginning, been pretty optimized. We take advantage of all sorts of features of the processors and storage and so on. So as you see here, Graph is actually faster than all of these genome analysis pipelines measured on a 72-core processor. And to our knowledge, Graph is the fastest software-based pipeline freely available to use today. Okay, so having established what graphs are and what they can bring to the table, I would also like to explore a bit the extra possibilities that are enabled when you use graph genomes. So, for example, there are three types of so there are many possibilities, and graphs can be either very general. We saw the example of one, the pan-genome graphs. They can be a bit more specific, as I mentioned a bit earlier, specific to a population, or they can be very specific, personalized graph, personalized to an individual or family. And finally, they could be minutely tailored to one purpose, wherein they are tailored towards detecting deleterious pathogenic mutations only. So we'll see examples of all of these. So first, population graph reference. These references capture the genetic diversity of specific populations. They enable more accurate read alignment and variant calling. And Seven Bridges provides custom graph construction services. Hopefully, Sarah will be able to talk about this in a short while. So this is the schematic of what a population graph is and how it uses the information. And in addition to the alleles, it also uses information about the prevalence of that allele and linkage and so on, and thus improves the cohort's variant calling accuracy. So we tested population graphs on several different populations, well, 3. And here we see the first panel. It shows how many extra variant calls you get as compared to the standard BWA GATK joint call workflows. We see that for European population, we get about 5% extra. For Qataris, a whole lot more. And for East Asian, something in the middle. And this is interesting. And this comes back to the point I was making right at the beginning about genetic diversity. Europeans are, as a population, Europeans are probably closest to the reference assembly that we have today. And East Asians are a bit more diverged. And Qataris, they do not feature in the assembly, and they do not feature in most data sources, like 1,000 genomes. So it is no surprise that a graph that is constructed from a Qatari cohort is improving the recall on Qatari specific variants significantly. And on the right, we see the drop in, or the improvement in read mapping that is enabled by using these graphs And I should say that the middle line, the blue line, is the results from the standard pan-genome graph that I was describing earlier. Whereas the green one is the results from the population-specific graph. So there is a substantial improvement in read mapping when using the standard pan-genome graph, but when we use population-specific graphs, we improve matters even further. And and just a quick aside, just in case you are wondering, that these extra variant calls that we identify on these cohorts with these graphs, are they reliable? So these are the QC metrics, and we see that they are well within the range of being acceptable, and so that indicates that the extra calls that we make are likely not false positives. And now let's turn to implementation of, or a more specific implementation of the graph reference, a family graph. And here, this graph contains information from only 3 genomes, and it is constructed by combining individual genotypes from family members, and once we have this graph, maybe it's better to see the workflow. So yeah, so this describes the workflow, the schematic above. We start with the samples, the parent samples, and we call them using the global graph or a population graph. And the variance that we get in the result, we use that to create a family graph or a parents graph. And then we map all the samples against this graph, where we expect that this graph has pretty much every single variant that is present in all 3 samples. And we call them and we combine them to get consistent output for the trio genotypes. And when we measure the Mendelian discordance between the call variants in it among the trios, we see that it is significantly lower than when the samples are simply joint called together, which implies that this genotype set is less likely to contain false positive calls, which in turn makes it easier to identify pathogenic variants of interest, be they de novo variants or biallelic recessive variants. And finally, another application of the personal graphs that we have been working on is using the graph genomes in order to analyze cancer samples. So, and here the idea again, the pipeline is described at the top, is that we take the normal sample and we analyze it using a population graph which is relevant to it. And we get a germline VCF, we use it to construct a reference, optionally augmenting it with driver mutations known to be relevant to the cancer being analyzed. And we get this cancer graph, and then we use it to map both tumor and normal samples, and this should make it easier to differentiate somatic variants from germline variants, because the germline variants would be part of the personal reference. So with this idea, we developed this tool we developed this workflow. It is still ongoing, but we have encouraging results. So on the left panel, you see that the graph. Left panel, you see the benchmarks on two samples, one real and one simulated. So the real sample comes from the well-known seq-C somatic benchmark. It's a breast cancer sample. And we, the same 50x tumor normal pair, we analyzed it with our workflow, and we analyzed it with Strelka2, and we analyzed it with Mutect. And we see that the round dots correspond to the results from the three pipelines on this. And we see that the graph leads to higher recall on this sample and marginally lower precision. And whereas on the simulation, on the synthetic sample, which is a HG003 and 004 mixture with HG004 as tumor, which is a pretty standard strategy for validating somatic pipelines. Again, we see that our pipeline again has higher recall than either Strelka or Mutect without any precision drop. So given that this is still a work in progress, we are happy with this. This is quite encouraging, and we hope to share more news with you on this soon. And in the right panel, we actually tried to estimate the significance of this extra recall. And here we have we studied a colorectal tumor sample, hypermutated, and this sample was published along with a list of mutations that were analyzed using a panel of variant callers in the protein coding regions. And there we see that, again, graph pipeline identifies most of the mutations, and happily, it identifies exactly the same number as Strelka and more than Mutect. But again, as I say, this is work in progress, and we hope to share more with you on this soon. There are some applications of graph genomes to somatic calling, which we are exploring. And yeah, so I think with that, I'm supposed to pass over to Sarhat.
Hi, Amit. Can you hear me?
Yes.
I apologize for the technical difficulty. For some reason, I lost all audio and video.
Okay. So can I stop sharing my screen, or do you want me to continue sharing?
I can start sharing my screen because I also do the demonstration at the end, so that would be faster.
Sure.
All right. So you see the infinite screen, and all right. So welcome everyone. That's a bit late, I guess, but I apologize again for the technical difficulty. But so in this last part of this presentation, I will walk you through what's graph-based solutions that we have available at Sembridges, and I'll discuss what will be available in the near future. So the first available solution that we have is our germline variant detection workflow. So this can be used for processing any type of germline data that you have. This workflow consists of our graph aligner and graph variant caller, both of which use our genome graphs, and a final stage of variant filtration at the end. So this pipeline is available on all of our platforms, as Amit mentioned. So if you are already a user of any of our platforms, you could immediately go to the public apps and copy this to your project and start processing your samples with this pipeline. To accompany this pipeline, we have also released our pan-genome graphs, both two versions of them, each one compatible with GRCH38 and another compatible with GRCH37 versions of references. And the pipeline itself is indeed compatible with many of the graphs that we produce, including population-specific graphs, disease-specific graphs, etc. And if you are an academic user in any of our academic platforms, again, this is completely free to use, accompanied by our pan-genome graphs. And I should also iterate that our tools are fully compatible with existing data formats. So the tools, the aligner, for example, takes in raw reads as an input in the form of a FASTQ file or even a CRAM or BAM file. And it produces alignments in BAM or CRAM format, whichever you prefer. The variant caller uses this alignment and produces variant calls in a file or a GVCF file, depending on what you want to do. And I should also mention that these VCF files are full of annotations compatible with what you would expect from commonly used variant callers, such as GTK's haplotype caller. And on top of that, we also incorporate additional graph-specific information, both in our alignments, in our BAM files, and also in our VCF files. And these can be further used to inform any tertiary analysis downstream. So we also provide some services at Sembridges. We can construct graph-based workflows for your specific application. We can construct custom graphs. They can be population-specific graphs, personal graphs, disease graphs, whatever you might think of. We already have some available population-specific graphs, namely European, Qatari, and East Asian, for which Amit already showed you the results. And we are about to finish the construction of our Pan-African graph. And all of our solutions, all of our workflows are implemented in CWL with Dockerized tools, so they are very easy to deploy to any computational infrastructure and run there. So what should you expect from us in the near future? First thing we're planning to release is the pathogen detection workflow. So this workflow utilizes a graph that contains the genomes of corona, influenza, and rhinoviruses, and multiple subtypes of those viruses. The aim of this workflow is to be able to identify which viruses are infecting a given patient, DNA data from a given patient, and it can do that very fast, and it can actually identify the composition of the infection, meaning that if the sample is infected by multiple viruses, it can readily detect that. The second thing we're planning to release is the TRIO workflow, and it again showed you some preliminary results on that. And by utilizing a family graph, we can make the variant calling very consistent across an entire pedigree compared to a linear approach or an approach that is based on a generic graph. And we are also working on somatic detection workflow that we intend to release. And as I mentioned, we are working on population-specific graphs, but of course, if requested, we can construct graphs for other populations than what's listed on the slide. And as an examples of, and we are working with collaborators on multiple of these projects. I'm happy to see that some of them already joined this webinar. And as examples of disease-related graphs, we are working on hypertrophic and dilated cardiomyopathy graphs. And all of these releases, of course, will accompany blog posts, white papers, technical documents, user guides to help anyone who is interested in using and utilizing these things. So with that, I just want to switch to our platform and quickly show you how you can use our graph germline pipeline very easily on our platform, and what methods are available to inspect the results coming out of this pipeline. So, and this is, I hope you're seeing this interface of our platform. So this is a project that I prepared before this presentation. It's called, so I named this for this presentation, and you are seeing the apps available in this project. So we're not interested in the other apps, but at the bottom you are seeing our pipeline, Genome pipeline that we have been discussing. And if I just click on that, you will see a detailed description of what the pipeline is, the methods, a very short summary of the methods used and the filtering used, and inputs that you should provide, and references, etc., and what the outputs will be like, and a screenshot of the pipeline. And going to the top of this page, I just click Run over here, and the platform generates a new task for me. And you can see the inputs on the left-hand side. There are a few inputs, but you might have already noticed that some of these inputs are already defined. So this is a feature of the platform. We are able to define recommended files for work language level, and these are recommended files. So the first one is the graph reference, and this file is the pangenome reference compatible with GRCH38 coordinates. It is in VCF format, so that it is very easy to manipulate, and users can look inside to what variants are available inside the graph and all that sort of stuff. And the intervals, we also have a set of intervals recommended to accompany this file, but this is for any whole genome sequencing sample. However, if you have a whole exome sample, or if you have targeted sequencing data, you can, of course, immediately replace this bed file with the regions of interest, the regions that you are interested in. And this, of course, linear reference is accompanying these two files. All you have to do is to define the reads for this pipeline, input raw reads. And I do that by clicking Select Files, and this screen will list all the available files in FASTQ format that you can use in the project. And there is only one sample in this project. This is the HU0022 sample, which is a genome in a bottle test sample. And it's, of course, there are two files because the data is paired, and I click on both of these files and click Say Selection to define my reads, and that's all I have to do. And I just hit Run and wait for platform to do the work. You can see that it says queued over here. It implies that this task has been submitted, and the back end of our platform is assigning an AWS instance, a computer, for this task to run. And as soon as this is assigned, you will see that now it's switched to running. You will see that the pipeline is running, and there are 3 stages in our pipeline, as I mentioned: alignment, variant calling, and filtration. You can also track that over here. Of course, we're not going to wait for the sample to finish. I pre-ran this task before the presentation, and I'll switch to that. Now this is a completed task with exactly the same inputs. You are seeing the outputs on the right-hand side. First, the alignments in BAM format, and the variant calls in VCF format. And you can see that the task took about 3 hours and 38 minutes. So this is a whole genome sample with an average of 35x coverage. So, and it cost a little bit less than $5 for the specific task. Now, I have also processed this sample using the linear pipeline, which is BWA-GTK pipeline. And I will show you a utility on our platform that you can use to compare the VCF outputs, variant calls, coming from 2 different pipelines. Or, I mean, VCFs, they can also come from the same pipeline with different versions, etc. This is a utility for you to compare variant calls against a truth set. This is a genome in a bottle set sample, as I mentioned. So this is the interface of our VCF benchmarking utility, and you are seeing two VCF files, each corresponding to a single row over here. The first one here is coming from the graph pipeline. The second one is from the BWA GATK pipeline. And the results are essentially precision recall, F measure for SNPs, indels, or combined. And you can clearly see that in all of these categories, graph pipeline is outperforming the linear approach. And another cool feature of this utility is that by clicking this drop-down list, you can select the regions in which you want benchmarking to be done. And for example, I can click this MHC regions, which is the region where we won the competition we mentioned at the beginning of the presentation. You can also see the F-scores. I'm just looking at the F-scores for sake of simplicity at this stage. And you can see that the difference between these two pipelines are much more dramatic. And especially for indels, the difference is even larger. And as you all know, indels are inherently more difficult to call compared to SNPs, and that's why we're seeing a larger difference here. So that's all I want to show on this interface. I'm not going to walk you through the entire set of panels over here. Now, this was about variant codes, but you might be wondering, if you wanted to visualize our alignments, the reads in the BAM file, what would you do? You could, of course, use tools, very commonly used tools such as IGV, Integrated Genome Viewer. However, since it's not graph aware, it's not ideal to visualize graph alignments because our reads are going through the edges in the graph, and we need a proper way of showing that. That's the reason why we have developed our own alignment visualizer, graph visualizer. So this is so you're looking at an interface for our cruncher on our platform. So this is still on our platform. You can spin up a cruncher, and in terminal you can do whatever computation you want, run scripts, or you can just run your IPython notebook, which is what we do almost every day to do our analysis on the cloud. And this is a Python package that we have developed. It can be used in the IPython notebook, but we also have a standalone desktop app that you can simply download and use as if it was IGV but graph aware, with all the interactive features that you would expect from a visualizer or a viewer. So this is available for everyone to look at. I'll just give you an example of an alignment here, so that I wouldn't take too much of your time. After loading your alignments and graph reference with a single line, you could plot a region of interest and using the alignments coming from these two pipelines. At the top, we're seeing the alignment from the graph pipeline. At the bottom, we are seeing the alignment from BWA MAM aligner. So the colorful line at the bottom is the reference, and the edges that you are seeing going on top of these linear sequence are the edges in our graph, in our pan-genome graph. In the top alignment, it's very clear that reads are going through this insertion edge indicated by the fact that they're going on top of this edge, whereas since BWA is not graph aware, it cannot utilize this edge, and therefore the coverage in this region is lower compared to the coverage that we get with the graph pipeline. Coverage is much more uniform in the graph aligner, and this is the utility, an example of the improvement that you would get by using a graph pipeline. So I believe that's all I wanted to show, and thank you all very much for joining our webinar and listening to our presentation about genome graphs. Please let us know if you have any questions in the chat box that you should be seeing on your screens. That's all, I guess.
Okay, so I've been reading the questions, and I think there are several very interesting questions here, and I will take them roughly in chronological order. So the first question is from Frank Bellman. Does it make sense to deploy region-specific graphs for alignment and variant calling? for example, focused analysis of disease locus? And the answer is absolutely. We actually have ongoing collaborations where we are looking at disease, hypercardiomyopathy to be exact, and we are in those cases we are making disease graphs which whose purpose is exactly as you say, the focused analysis of uh, disease causing gene regions. Um, so yeah, I hope that answers your question. If it doesn't, feel free to put a follow-up in the chat window. Next question is from Bryce Sarver. Any thoughts on performance relative to Illumina's new Dragon offering? Yes. I think it is important to recognize that the Dragon is a hybrid software and hardware solution, and they benefit a lot from the hardware acceleration. So, and I'm aware that they can do a whole genome, analyze a whole genome in 20 minutes or less. So, so being a software solution, we can't approach that, but our vision for graph references is that we want to go beyond alignment simply. So we want to incorporate more information in the references, which will help identify variants more accurately. For example, allele frequencies and linkages in cohorts, phasing in trio graphs, and so on. Do you want to add anything to that, Sarath?
Not really, but I think you explained it very well. I just want to say maybe that where we want to improve the capabilities of the current bioinformatics toolkits is not the speed and computational efficiency, but providing more value in the sense that you get more information from your data by using a graph genome, rather than claiming that you can get the information in a few minutes.
Yeah. Okay, this next question is also from Bryce. Have you used this, presumably meaning graph genomes, to look at situations where there are CNV alleles at different frequencies in large populations? For example, could a graph be constructed that would be able to incorporate a specific CNV or identify one with more confidence when dealing with population scale cohorts, especially with short read data? Yes, so as far as the current released graphs are concerned, we are able to do that. As for CNVs specifically, and this functionality that you mentioned, the next update that we are working on for our graph genomes, they would have support for this. And hopefully we will be able to present good benchmarks showing the difference in CNV calling that is afforded by using prior information. Next question is from Durga Adipalli. Sorry, I joined late. Do the graph workflows use GPUs? And if yes, can a user configure the GPU usage? So we don't use GPUs because the read alignment problem is not, or variant calling problems are not GPU friendly. And if you look at the paper, our paper, the link is in this slide deck somewhere. You will see that the algorithm that we use is a seed and extend algorithm, and and we use hashing of the reads to find the seeds. So that problem is not inherently GPU friendly, and I believe if you want to use hardware for breed mapping, then the way to do is what Dragon does, but that's a whole different kettle of fish.
I just want to add that even though we use CPUs, we can use at the workflow level some parallelization optimizations so that the task I show, for example, was close to 4 hours, but we can reduce the runtime for a whole genome, 50x whole genome sample under 2 hours, if need be.
Yeah. Okay, the next question is from Alessandro Bornello. While you implement specific versions of Graph, how much can we use the general version for each of these tasks? So, okay, that's an interesting question. And I believe I had a slide that was showing that when we compared the performance from population-specific versus the general graph. So in this plot, in this figure, you see that the blue lines are the gains that you get with respect to the standard BWA workflow when using the general graph, but you get additional gains when you use the graphs that are tailored to the task. So yeah, and that's why constructing specific graphs is one of the services that our team provides. I hope that answered your question. Frank Pullman, are you exporting phased VCFs as the default? Is your variant caller phase aware? Not in this version, unfortunately, but it is an enhancement that is, it is on our roadmap. That's as far as I can tell you at this point. Okay, Mark Adams, for examining genome diversity, how could using a pan-genome graph compare to a tree sequence format like TSKit? I am, maybe Sarah, do you want to take this one? Your team had some work on the usage of the graph, like edges covered and so on, right?
Mm-hmm. I don't quite understand the question. Could you repeat that? I'm not familiar with TSKit.
For examining genome diversity, how could using a pan-genome graph compare to tree sequence format? Okay, I will take this. So I think what the questioner is wondering about is that if you map a sample using a graph genome, can you use that mapping information to identify the subpopulation that a sample comes from, as you do with phylogenetic trees? Or if two samples are aligned against the graph, then can you determine if they are from proximate populations? And I would say that it's an interesting idea. It is certainly possible in theory, but we have not got any script or any tools to do that yet. But I am making a note of it as we speak, and I'm sure we should be doing this. Okay, next question from Sinem. How do you decide the optimum population size to build a graph reference?
Should I take that, Amit?
Please.
So we have optimal size for a population graph. So we have a method to measure the genetic nucleotide diversity of a population. There are already many methods published in the literature. We tested a few of those, and we can measure the diversity within a population. And depending on how diverse the population is, we will need, of course, more samples. And the actual number of samples that we need to pick is coming from empirical results that we have tested on actually the population that you are seeing right now on the screen, and also on other populations. And we tested this on databases such as 1000 Genomes and Nomad to see if we are covering enough of the genetic diversity of a population so that we provide enough information for our aligner and variant caller. So we have done all these tests, and we have developed we have a finalized method that you could be able to just plug and play. Given your samples, we can immediately test a subset of the population and say that, all right, we need like I'm just making up this number, but we might need like 1,000 samples to cover the 85% diversity of this population. We can come up with these numbers easily.
Great. Next question is, what are the main differences between graph workflow and other graph-based tools? I think you should take this one.
Other graph-based tools being, I assume, not our tools, but other tools such as VG that you presented a slide for comparison. So then I believe one of the most important differences is that our tools are very scalable compared to other ones, and the reason is that we are optimizing computationally our tools so that they use a reasonable amount of resources, even resources less than other linear-based pipelines, such as the ones you are seeing on the screen. And therefore they're scalable on the cloud, especially. And our tools are, I believe, that they're mature enough so that they can be reliably and consistently be used on large-scale projects, which we have proven on a few of our collaborations until today, and we hope to extend our portfolio through our collaborators. But I'm not going to go into detail of how the other tools work, but from my experience, I just can say that they're extremely resource hungry and performance suffers depending on how you use the tools. It's very difficult to use, I would say.
Yeah. And to that, I think I would add that one important differentiator is that our tools use the standard file formats throughout. So our read mapper generates a BAM file with extra information in it, which is consumed by our variant caller, which generates a VCF file, and you can supply a bed file, etc. So at any stage, you can check your work or replace the tools like we showed with Manta running on graph-generated BAM files. And there is no conversion step involved. So I think this is one difference, which might be significant for many users. And also, I would like to stress that our view of the graph reference is more holistic than just being a scaffold for read mapping. And I am not aware if the other graph-based workflows are working towards this vision or not, but I haven't seen any evidence. So that could be a difference also. Thank you for the question. Okay, we have a couple more questions left. One is, how does performance scale with graph complexity?
So I can comment at the workflow level, and maybe afterwards you can comment on the tool level, maybe, Amit. But at the workflow level, I would say it scales well. The complexity, unless you specifically make a very short region extremely crowded, and your data is coming from that region, your alignment data, the computational resources are, I would say, the increase in the computational resources would be minimal. And I can say, for example, for a whole genome sample on an instance that with 72 cores, the memory usage is about 50 gigabytes. And if I double the size, if I don't know, increase the size, graph size by 50%, our pangenome graph by 50%, it would go up to 70 gigabytes, something like that. So still very much within the resources available, readily available on our platform or any cloud service.
Yeah, and I think I would only, from like a technical point of view, I think the first slide that I presented about the alt config and how we add it to the graph, you would immediately see that the processing that this representation needs is much less if you were processing the alt sequence as well as the reference sequence. So this is one of the strategies that means that we handle complexity quite well, and the algorithms that we use internally, they are all scalable. So yeah. Okay, next question. Do you provide private software for the user to make their own graph?
We do. So it is not. So it is available. We can allow user to make his own graph, or we can provide this as a service. However, this is not available as if you are interested in, let me pull up the slide for contact. So if you're interested in making a custom graph on your own or with our collaboration, please feel free to contact us. So we are indeed, in all of our collaborations, we are using custom graphs. And these are done as case studies to show that graph genomes are useful in different genomic contexts. So that's one option. But if you're commercially interested in making your own graph with our tools, I believe that it's better to contact us, and then we can get in touch with you and discuss what options are available. But those services are available. Tools are available.
Cool. Okay, I think we are running out of time. So one last question. Asif Chinwala asks, how have you thought about how to integrate other omics data into the graph? Each, for example, RNA-seq has a longitudinal view of expression across the alleles. And Asif, the answer is yes. We use this same strategy in the pathogen graph or the respiratory virus graph that Sarah mentioned. In that, our strategy is essentially to store abundance level with each allele, so that once you map your sample, you not only get the variance, but you also get an assessment of the species or lineage abundance. So this is certainly possible, and we have explored this. So yeah, I think so. We will close now. Thank you very much for joining. And if you have any more questions, please feel free to contact us. If you want to collaborate with us, we are always seeking out new sources of data and collaborations. And it has been great. Thank you.
Thank you all very much.